Hello Psyc100 students, I'm your instructor, Carrie Kerstetter. I wanted to do a brief introductory video to tell you a little bit about myself, as well as the syllabus, go over some expectations, make sure that we're all on the same page. And then I'll go into a little bit about psychology as a science, because that's the first topic that you guys are going to have a quiz on in just a couple days on Thursday. You have your first quiz. So my name's Carrie, again, and I am a postdoctoral researcher at Seattle Children's Research Institute. I got my PhD back in 2011 at UC Santa Barbara in psychology, and currently I do research on the neural pathways related to addiction behaviors. So a lot of experimental basic science research, pretty relevant to what you're learning about this week as far as experimental design and all of the different experimental designs that you can have, and the caveats to all of the designs, the advantages and disadvantages. The Canvas system I think is pretty slick. I think it's working pretty well. If you have any issues with it, please let me know. You can email me at my South Seattle email, or use the Canvas system to email me at the top right corner of your Canvas screen. Today I'm going to talk a little bit about psychology as a science, but I wanted to first talk about the course syllabus. So I've organized the course to kind of go with a Tuesday-Thursday theme of assessments. So you're going to have a discussion or a quiz once or twice a week on every topic. So you're going to be evaluated as we go for each topic that we cover. So either you're going to have a discussion, quiz, or sometimes you'll have both for a given topic. So please go over the calendar and get a sense of the workload, the timing, and if you feel like this is definitely something that you can do, because the syllabus is kind of an agreement between you and I, saying like this is what's going to be happening this quarter, and by staying in the course you're letting me know like, yes, I can definitely handle this, I'm excited, and I'm going to be going through all of the modules as we go along and be able to complete all assignments on time. So I really appreciate it if you guys go through the entire syllabus and make sure that everything makes sense. Please send me an email if something doesn't make sense, and just know that we're all on the same page. All right, so that was a little bit about me, and now I'm going to talk a little bit about psychology as a science. All scientific research begins with a theory. A theory is a general, tentative explanation that tries to explain diverse findings on the relationship between variables, and therefore a theory can inspire a lot of different veins of research. So it's not a specific question, it's just a general theory about how things work that can lead to a lot of specific hypotheses or research questions. It can inspire a lot of research. And theories are very important. Novel theories are very important, and it's always exciting when a new theory is proposed and then it inspires a ton of new research. Perhaps one of the most well-known theorists in psychology is Sigmund Freud. Sigmund Freud had a lot of different theories about development and personality, and they were extremely interesting, especially for the time. His theory of development, such that he believed that the libido was the greatest motivation that we had, and that led to a lot of crisis that we had to get through in order to achieve successful development, was extremely interesting. Now, he didn't necessarily have specific hypotheses to test in experimental design, but it inspired a lot of future research because it had such interesting theory. So as you can imagine, with Freud's theory about the ego and superego, the id, the psychosexual development, oral fixation, all of that, he didn't have a lot of experimental design in his theories. However, it did inspire a lot of research. To get to a specific research question, we actually need to come up with a hypothesis. A hypothesis, as opposed to a theory, is a specific prediction about the relationship between two or more variables that we can test using experimental design. And there are a variety, as you can imagine, experimental designs. There are several research designs that you can use if you want to conduct a research experiment. Perhaps the easiest one you could do is a descriptive study. So this includes case studies, natural observation. You could do a correlational study if you're interested in how two variables are related, or you could do an experimental study where you actually control variables, control the groups. One example of descriptive research is Freud, who conducted case studies, or Jane Goodall's natural observation research on chimpanzees. I'm sure you guys are familiar with Jane Goodall. She did very famous research for decades where she lived with chimpanzees in their natural environment and took notes on them for decades, really understanding them as their own population. And what she found was that they not only used tools, but they made tools to use for specific purposes. They also had highly sophisticated social structures and would even have wars for years between different social groups. So things that we think of as human are more high-end on the spectrum of behaviors that are only specific to us. She actually found were also there for the chimpanzees. So the main take-home point for descriptive research is basically examining the world, either individuals or groups, as they are, and taking a snapshot of what the world looks like and just describing it. I'm going to talk about correlation. Correlation research seeks to understand if two variables are associated, if they're related in any way. So a famous correlational experiment was done in Stanford in the 1960s called the marshmallow experiment. This experiment was about delayed gratification. So what they did is they took young children, I think between the ages of 3 and 6, and they offered them either a small reward now, so they could either have 1 marshmallow right now, or they could have 2 marshmallows if they could wait 15 minutes. So they let children decide. And then, much later, they wanted to see how those children were doing. So in adulthood, so they were about 18 when they looked at these kids again, they found that the children who chose the later, bigger rewards, the kids that were okay with having delayed gratification, could wait out for the bigger reward, had better achievements. So one of the measures they used was SAT scores. They found that delayed gratification was associated with higher SAT scores. So those are two variables that are associated. They're correlated together. If someone is more likely, or if someone is the type of person that is okay waiting for a bigger reward rather than being more impulsive and taking the reward that's right in front of them now, even though they know it's smaller, then perhaps they will do better in life down the road. Now, it's really important to note here that correlation is not causation. This study is not saying that because they chose the 2 marshmallows, because they chose to wait for 2 marshmallows, that is why they got higher SAT scores. You can't say that at all. All you can say is that these 2 variables are correlated. They're positively correlated, meaning that the kids who would wait had the higher SAT scores. And that's pretty much it. You can't say anything more than that. So that's a big disadvantage of a correlational study. A lot of basic research. So basic research is research for the sake of knowledge itself, just to know more. So a lot of brain research is done, or biology research is done just to know more about the cells of the body. So biopsychologists do a lot of basic research just to find out more about the neural populations in your brain, figure out which pathways are connected to other pathways. So are we connected from our PFC to our, are we connected from the prefrontal cortex to our midbrains? Which pathways are going in between the midbrain? Biopsychologists like myself are really interested in all of those little details. Like, what exactly is going on? Can we just figure out what these are? It's not necessarily about, okay, well, how do we treat a disease? How do we give this drug, and what's the impact for this disorder? A lot of times we're doing basic research just to know more about our systems, just to know more about our mental processes as well as our behavior, because they are directly linked. So a lot of basic research is experimental. As you can imagine, the research that I do is experimental. And experimental research means that we are controlling the groups and we are controlling the independent variable. So the critical thing in experimental research is that we are randomly assigning subjects into either you're going to get the drug or you're going to get the placebo. And that's done randomly so that the groups are equal at the onset of the experiment. And then we get to decide what is the independent variable. What are the different levels? So let's say this group of people are getting a drug that we're interested in measuring the effect, and these people are getting the placebo, and then we get to choose, okay, what are the doses of drug that we're going to get? That's our independent variable. What are our different drug doses? And then we have our dependent variable, which is going to be the behavioral measures. So let's say we're looking at drugs for depression, and we're giving all of these antidepressant levels, these different doses of the drug, and then we have the other subjects that are getting the placebo. Our dependent measure will be their clinical effect. Are they having any improvement in their mood and their affect due to these different levels of the antidepressant versus the placebo group? So that's an actual experiment because we're randomly assigning groups, and we're saying that one group is going to get the drug, and that's either going to lead to a mental or behavioral impact or it isn't. And then we can say whether or not it's caused by the drug. So that's the advantage of doing an experimental design. We can say that something is causal rather than just related with the correlational studies. An additional experimental design is the quasi-experimental design. The difference between an experimental design and a quasi-experimental design is that the quasi-experimental design does not randomly assign your subjects into groups. So you can take a population of people where you have some people taking one antidepressant and you have another set of people who are taking a different antidepressant, and then you can measure the clinical effects in those two populations, in those two groups. You haven't randomly assigned them to the groups, but essentially they're already in groups, and you can take the clinical effectiveness of those two drugs and see, which one is doing better and which one has greater side effects and that kind of thing. So that has some advantages. The data is already there, essentially. The subjects are already in the experiment. However, the disadvantage is that you could have some confounding variables. You have less control over the population, over the sample. So you really have to think critically about your behavioral measures. And are they purely because of the difference in your independent variable, the drug? Is it due to the independent variable, or is it due to some other confounding factor that is due to these two groups being significantly different in some other way? So I'm going to end the video here. There are many other topics that are covered in the online module for psychology science. You're responsible for all of the online content, so please read through that thoroughly for your quiz this Thursday, and I will see you guys online very soon. Be well.